this video is the cell cycle regulation of cyclin CDK by APC, Wee1 and CDC25. This video has two examples of balance, or in other words, regulation. One of the examples we're going to talk about is how the amount of protein is regulated. The other example is going to talk about how we look at how the amount of active protein versus inactive protein is regulated. And so I used a balance here to think about those two processes and how they can modulate back and forth. So the first one we're going to talk about is the amount of protein, in other words, the concentration of the protein, and the example we're going to use is a cyclin. Because these are general processes, this can actually be applied to any protein in the cell, and it can also be applied to any cyclin in the cell. We've seen before how important the amount of cyclin is to determine where the cell is in the cell cycle. And as a reminder of that, we've got this shenanigans going on here, looking at the concentration of S-cyclin through the cell cycle, and then we also see the concentration of M-cyclin through the cell cycle. So the two things that determine how much protein is present in a cell at a given time are, first off, its production rate, and secondly, that particular protein's degradation rate. In other words, proteolysis. The production rate is largely a function of the transcription because transcription will determine the amount of translation. So both of these things need to happen to produce proteins. And the other thing that needs to happen to determine how much protein is present for a given protein at a time is to consider its degradation rate. And so the degradation rate can vary, likewise the production rate can vary. Degradation rate, again, is about proteolysis. Proteolysis is proteolysis, so the breakdown of proteins, in particular breakdown of the protein polymers, or a protein polymer, into its amino acid monomers. And so this is basically like recycling. Proteolysis occurs by enzymes called proteases. So again, ACE being enzyme and proteo being protein, so this is breakdown of proteins. And your book highlights a particular mechanism in which there's a collection of proteases that form something called a proteasome. It's a protein complex. And this particular protein complex, the proteasome, is brought to a particular protein by certain tags that are added to that protein. And so these tags, I'll talk more about soon, but include ubiquitin, and they are essentially a big neon sign that is targeting this protein complex or other proteases to come and recycle it. And so proteins can have a variety of other tags that indicate their expiration date. Some proteins are meant to be maintained longer in the cell, and so have some kind of protective mechanisms on them. Other ones have tags to destroy them more quickly. And so we can think about this balance of production and degradation for any one protein. And so there's just a kind of a balance between the two of these. And so if we have more production going on, that means we're going to have higher levels of the protein. And if we have more degradation going on, then we'll have lower levels of the protein or lower concentrations. So to think about what's going on with these two different processes of production and degradation, I want you guys to think about the relative speed of what you would predict for each of these, thinking about where in the cell transcription in particular takes place and where degradation would take place. And so basically, as you may recall, when we have genomic events taking place, genomic events which are in the nucleus, things like specifically transcription, that's relatively slow. And we put a number to that as taking hours to occur. On the other hand, cytoplasmic events, which that's what where the proteases and the proteasome are found, cytoplasmic events are relatively quick, on the order of seconds. And so this is a reason why for many of the cyclins we see that there's this gradual increase up. That's from this slow process of transcription and then thereby translation. And then on the tail side here, when we see degradation, it goes a lot more quickly, and so we see this rapid drop-off due to this cytoplasmic event. This is figure 18-9 in your textbook. Here we're looking at a specific example about a cyclin which is being targeted for degradation through ubiquitin. So ubiquitin is a small protein, and it's a common covalent modification to proteins. This is typically a short chain of ubiquitin proteins that's added to another protein. And so you can see that over here, you've got a ubiquitin chain, so each of these little red squiggles would be a ubiquitin protein. Notice that they're all covalently bound to each other, since you can see a solid line there, and it's also covalently bonded to this cyclin because of that line. The ubiquitin is added by a protein complex called APC, that's the anaphase promoting complex. And that's not drawn in here, but I just did. So there's our APC, anaphase promoting complex. It adds ubiquitin to particular cyclins. And then while this cyclin is hanging out in the cytoplasm, then the proteasome is recruited to that area due to the ubiquitin chain. And then the proteasome will destroy, in other words, to dismantle the cyclin protein into its monomers. It just kind of looks like little blobs here, but it'll recycle it into monomers and cause that protein level to drop. Likewise, this will then lead to the CDK being inactive because it needs its partner, cyclin, to be bound to it. Now let's take a look at this summary of the cell cycle and see if we can figure out which of the cyclins is drawn here. So it's not labeled, this is just setting, it's a kind of generic cyclin, but we can actually put a specific name to it. If you look over here, so the APC is up here at the M phase, and you can see in my little note here that that leads to the degradation of cyclin B. So let's add that on here, cyclin B. Also, let's think a little bit about the name, anaphase promoting complex. So it's going to promote anaphase. Anaphase is part of mitosis. It's at the tail end of mitosis over here following metaphase. And basically, this protein complex is going to help move the cell cycle through to that point. Now, we're looking at the tail end of the activity of cyclin B. Notice that it's actually part of the checkpoint previous, which is of the G2 to M transition. And so it's CDK now. We could also label that CDK1. And these are the mammalian names that I'm using here. And to be complete, I've added the names for the Xenopus versions. So it would be MCDK and M cyclin. And again, we're looking at the tail end of their activity. Now let's rewind a little bit and talk about how we ended up with these partners working at the particular checkpoint. This figure shows the names of the Xenopus version, but I wrote in the version for mammalian, so cyclin B is the cyclin and CDK1 is the CDK that corresponds for this particular checkpoint. And we'll talk some more about that checkpoint soon, but what I want to highlight here is this is a take-home message thinking about regulation and how we need to use two different regulators to get this process just right, the Goldilocks condition. So this is an example of how we're looking at the balance for the amount of active protein. And what we're looking at here for balance is looking at how we balance the amount of active form of the protein versus the amount of the inactive form. And the answer is that there are two enzymes which control this. One of them is called Wee1, and this is an inhibitory kinase. The other enzyme is CDC25, and this is an activating phosphatase. So each of these is shown on the pathway above, and they've got their own little shapes, and we're just checking out that each of these enzymes is active. And so I didn't put their little red lines, but basically the Wee1 is up here. And basically we've got the mitotic CDK and the M cyclin, and when the inhibitory kinase Wee1 is active, it adds a phosphate to the CDK, and that inhibits the activity of this pair. Now normally this pair is going to control the G2 to M checkpoint, and so this means that these guys are kind of in a holding pattern when Wee1 will add this phosphate. This is also one of the few examples we've had so far of an inhibitory phosphate. And this seems very weird, but just hang in there with me. So this basically gets everything up at the ready. We immediately disable it just to kind of put it in this holding pattern. And then when the time is right, then CDC25, the other enzyme that is in its active form, will remove that exact phosphate, and thereby that means that now we have an active MCDK, which can allow the progression of G2 to M. And again, this seems very weird, like why add a phosphate just so you can remove it later? But remember that we're looking for some very fine tuning and hitting this balance. It is much quicker for a cell to add and or remove a phosphate than to go ahead and destroy this cyclin and then wait for hours until that cyclin comes back. So this is a really quick way to get some fast results. And that's a key process for the cell. Something else I want to introduce you to is a way that we describe the regulation of that each of these enzymes is imparting. So there's positive regulation and there's negative regulation. And basically negative regulation is when we have an inhibitor. So Wee1 is a negative regulator of cyclin, whereas CDC25 is a positive regulator since it's an activator. And if we think about this in terms of the analogy that I used for controlling the speed of your vehicle, or kind of the activity level of your vehicle, I talked about a brake and an accelerator. And so the gas pedal or accelerator, that is the positive control, whereas the brake is the negative control. And so basically for the cell to hit this G2 to M checkpoint just right, then we've got our negative regulator, Wee1 as the brake, and then we have CDC25 as the positive regulator or activator of CDK, and in other words, for this progression from G2 to M. Now I want to remind you about this figure, figure 18-5, as well as our cell cycle kind of summary, and how this process fits in, and both of these balance points we've talked about. So we just talked about how cyclin B/CDK1, in other words, the M cyclin and M CDK, are used and regulated by Wee1 and CDC25. So that's labeled here. And as a reminder, this figure here shows us that progression. Remember that the M cyclin is going to build up slowly, and then when it hits kind of this critical threshold, then MCDK will be at its high activity level, and that's going to happen through mitosis. So that basically is going on right here at G2 to M, and then it's going to continue until APC comes in and adds the ubiquitin, and then that leads to the degradation of cyclin B. And so then that is what we see right here is APC's activity that leads to the crash of the cyclin levels and therefore also the crash of the CDK levels. One thing you could think of here for why we don't see this buildup, kind of slow buildup of MCDK activity, there's a lot of reasons why that could happen. One is the threshold, but the other is due to this series of events here, that we've got CDK that's held in kind of a holding pattern with this inhibitory phosphate, and then it all gets activated very quickly through the CDC25. And a lot of that has to do with the regulation of these enzymes, which is something we're not going to talk about.